Before going into the practical of plasmolysis, let's have some theoretical knowledge about plasmolysis. Plasmolysis is the shrinkage of the cell membrane away from the cell wall due to the exoosmosis. Now student, we will discuss that how the exoosmosis of the cell is taking place. When a cell is kept inside a hypertonic solution, then what happens? The solvent, that is the water molecules, will travel from its higher concentration to its lower concentration. As a result, water comes out of the cell and it releases into the hypertonic solution. And this leads to the shrinkage of the cell structure. This condition is known as plasmolysis and the cell is known as plasmolysed. Now student, we will see how this hypertonic solution is helping in the plasmolysis process. For this experiment, we are selecting Rhoeo leaf, or we can continue this experiment with onion peel also. But for our convenience, we are using the Rhoeo leaf. Rhoeo leaf is a variegated leaf. If you see the leaf, it has two colors. On the upper surface, it has green color and on the lower surface, it has purple color. So the question arises, why we are selecting Rhoeo leaf? Not we are selecting any other green leaves. The reason is that student, we have to see the plasmolysed cell under the microscope. So in that observation, the chlorophyll will create hindrance in observing the cellular structure. That is why we are selecting the purplish epidermal layer of the Rhoeo leaf so that we can easily see the cells under the microscope. The material required for this experiment are the Rhoeo leaf, forceps, slide, cover slip, compound microscope, needle, tube, sodium chloride of 2 concentration. Here sodium chloride 0.1% concentration is used as the control set and 5% will act as the experimental set. When you are going to take off the peel from the lower epidermal layer of the Rhoeo leaf, one piece should be dipped in the 0.1% of NaCl. That will act as the control set where the concentration of the solvent is more, and another peel will be kept inside the 5% NaCl, which will act as the hypertonic solution. After keeping this whole setup for 20 to 30 minutes, we will observe it under the microscope by covering it with cover slip. Here we will see that the 0.1% solution of sodium chloride will act as the control set where no plasmolysis can be seen, whereas the 5% sodium chloride is showing the plasmolysed cell. So from here we can conclude that 5% sodium chloride is acting as the hypertonic solution, which leads to the exoosmosis of water from the cell. As a result, the cell membrane is shrinking away from the cell wall and it is becoming plasmolysed by localizing at one corner of the cell. As you can see that the white portion are more in plasmolysed cell, and in case of control set, the cell membrane, they are stick to the cell wall. I hope student, the concept of this experiment is clear to you. Now let's see some of the precautions we should keep in mind while performing this experiment.